In our study, we aim to understand the molecular mechanisms that are responsible for triggering cachexia during chronic virus infection.
One value of our study is that it provides an infection model for the research community to study the mechanisms of cachexia. Many patients suffering from cancer, AIDS, or chronic inflammatory diseases have one mysterious condition in common that is called cachexia. In our recent study, together with collaborations from the University of Graz, the Medical University of Vienna, as well as colleagues from Germany, Switzerland, and the United States, we elucidated a novel mechanism of how chronic viral infections cause cachexia. Cachexia occurs as an unintentional weight loss due to reduced food intake and altered metabolism. And patients lose their strength due to the depletion of their fat and skeletal muscle mass, which cannot be reversed by nutritional supplementation. And we know relatively little about cachexia in infectious diseases actually, but the majority of advanced cancers show signs of cachexia, which is thought to be involved in the premature death of the affected patients. And despite this huge clinical importance of cachexia, there's still no effective treatment available against this disease.
In our study, we tried to understand the molecular mechanisms that are responsible for triggering cachexia during chronic virus infection. And so we did that in a well-controlled infection model. When we look at the immune triggers to try and understand which elements might be responsible for triggering cachexia, we found that, to our surprise and contrary to our expectations, the trigger was mainly CD8 killer T cells. In order for these cells to trigger cachexia, they needed to receive activation signals from the antiviral cytokines type 1 interferon, and they had to recognize the virus. We further characterized the effects of cachexia on behavior, physiology, and metabolism, and we found that the onset was associated with a reduction in eating, drinking, activity, and altered metabolism. More specifically, we found very interesting changes in the fat tissue and were able to demonstrate that the infection led to the activation of complex molecular cascades that led to the depletion of the fat depots and the release of fatty acids into the circulation. We also found severe reorganization of the fat tissue architecture that we think might provide key to a better understanding of cachexia.
One value of our study is that it provides an infection model for the research community to study the mechanisms of cachexia.
One of the most surprising findings of this study is the role of CD8 T cells in causing cachexia. And further studies will need to be done to understand whether similar mechanisms are involved in other infections and in cancer.
Exactly. In addition, we're convinced that a comparison of cachexia in both infections and cancer will provide very valuable and unprecedented molecular insights. This may help us to understand whether cachexia may possibly even provide an advantage to the host during infection. Ultimately, we hope that this study will contribute to renewed efforts for developing innovative therapies that prevent or at least slow down this mysterious disease called cachexia.